The beauty in cyanobacteria is that it starts with nothing.
Photosynthesis is the most unique process in nature.
We hope that we can, I would say, revolutionize the solid-state cell factories.
Around 2 billion years ago, something extraordinary emerged from the primordial seas of our planet: photosynthetic bacteria. These microorganisms generated the Big Bang of life by creating a breathable atmosphere above sea level.
Photosynthesis provides us with molecular oxygen, with food, and also with energy.
Thanks to decades of biotechnological development, this unique power can now be harnessed by employing the organisms to produce chemical compounds that we need in our everyday life, ranging from pure hydrogen to biofuels and commodity chemicals, without the need of any fossil raw material or complex carbon sources.
They don't need sugars to grow. They produce biomass from sun energy, most abundant energy, and that's why they are also called primary producers.
Algae biomass and fatty acids have already been cultivated for food and fuel, but these are traditionally grown in dilute suspension cultures, which has its drawbacks.
One of the major limitations of suspension cultures is their low light utilization efficiency. In the dense algal cultures, we have a significant problem with low light utilization efficiency, with the self-shading effect.
One of the major problem in upscaling this type of light-driven reactions is, yes, if you make the reactors very big, then the light transmission really decreases. And so, yes, the cells that are on the side gets illuminated, but then you have a huge amount of cells inside that doesn't get illuminated.
The FuturoLEAF project tackles this problem with a unique bio-inspired approach by mimicking the green leaf. Instead of a suspension culture, the cells are embedded into a solid, thin layer cultivation platform.
If we are able to embed or bind the cells into these thin matrix layers made from, for example, nanocellulose gels, we are able to overcome most of these bottlenecks.
One of the advantages of the solid-state cell factories is significantly increasing cell density, which gives the high volumetric production capacity to the production system.
We are able to increase the light utilization efficiency. We are able to reduce the water consumption, the energy consumption.
Another benefit is the life cycle of the cells. The researchers at the University of Turku have been able to keep the cells alive for over 200 days. Material scientists at VTT and Aalto University in Finland have developed the leaf-like solid-state matrix.
We want to offer those cells a supportive material that creates an environment like in that green leaf.
Our intention is not just blend cells with nanocellulose, but to look at these light management aspects, skeleton aspects, and water and nutrient transport aspects, and try to build up the architecture in a similar fashion as it is in green leaves.
The matrix itself is made with nanocellulose, a tunable gel-like material with strong water interaction properties.
And why nanocellulose plays here the key role is that it is not polymeric, it is fibrillar.
The cellulose nanofibrils, they are like long entangled network, and this network is cross-linked either covalently or ionically.
We're working with tempo oxidized cellulose nanofibers as the matrix material, and even though it has some very good benefits in terms of the immobilization application such as transparency, but tempo CNF is not stable in water as itself, so we need some kind of strategy to improve the water stability.
And this is something we can really utilize in this cell factory concept. You know, for instance, if the cells, if they are excreting a product, you know, a chemical, then the amount of water and its viscosity, things, I mean, the properties of water there, which are dictated by the cellulose-water interactions, they control how it diffuses out of the matrix or even inside the matrix, things like that. The light response is partially governed by that. So this is what we are really working. We are really working on fundamental interactions.
One of the benefits of bio-based nanomaterials, like nanocellulose, is that they are tunable and versatile and can be tailored to the specific needs of the different cells and end products. In order to fine-tune the passive relationship between the photosynthetic cells and the matrix, the research institute CNRS in France is exploring the use of molecularly imprinted polymers, so-called MIPs, to actively attach the cells to the matrix.
We are working on materials that are called molecularly imprinted polymers. They are also known as antibody mimics, and like biological antibodies, they can recognize and bind very specifically other molecules, proteins and smaller molecules. And that is what we do. We develop these materials against surface proteins of photosynthetic cells, and we will then use them combined with the nanocellulose support matrix in order to sequester these cells in the artificial leaf structure.
The main aim is to find a solution that it doesn't cost a lot, it's cheap, because the final target of the project is to be in the scale up the bioreactor and the cells production.
By using the MIPs, the team has been able to attach the cells to the matrix and control the location of the cells in the system. Apart from designing the matrix, the photosynthetic cells themselves must be designed to produce the target's end products.
We need to engineer cyanobacteria and cells. We use synthetic biology approach for this one and force them to produce the compounds, chemicals we are interested.
Engineering photosynthetic cells is highly complex.
It is not like you engineer one gene, you strike one gene and you get a growth. It's like improving milk, like from a cow or so, or increasing a grain weight. It is a multiple gene factor.
Researchers at the Graz University of Technology in Austria and the University of Turku have demonstrated the production of a plastic precursor with genetically modified microalgae.
The targeted compound that these cells are producing is epsilon caprolactone, and it is used as a polymer precursor at the moment. And it typically is made in an unsustainable way, but this way how we are making it with the photosynthetic cells, it would be sustainable.
The next challenge is to scale up production.
Also look at the bigger scale, how we can move it from only like these so big vials or flasks to a really a bigger bioreactor, which is then probably also more interesting for industry.
While the field is still young and requires extensive fundamental research, the opportunities of harnessing microalgae are vast. The FuturoLEAF project has partnered with Simris Biologics to explore the commercialization of solid-state cell culture technology.
Yeah, I think today the FuturoLEAF concept is probably not applicable for the production of low-value products like biofuels or chemical feedstocks, because here there is still quite some process optimization and down engineering required towards higher production yields and lower production costs. However, I think that already today the FuturoLEAF concept is applicable for the production of higher value products, like especially pharmaceuticals, but also specialty chemicals. And this counts especially because of the highly controllable and reproducible cultivation conditions the FuturoLEAF concept allows for.
The multidisciplinary work of the FuturoLEAF project has generated exceptional results, redefining how sustainable algal cell cultures could look in the future.